Welcome. In this lecture, we discuss specific responses by the immune system, and we will expand some of the basic models we have studied so far to incorporate some of these specific responses. There is plenty of references for this lecture you'd like to perhaps review very carefully. Chapter 42 on the book OpenStax College Biology. Chapter 42 has to do with the immune system. Is an excellent reference for the content that we discuss here. So let's review some terminology. There's something called the antigen. An antigen is a substance that is capable of inducing the generation of a specific response by the immune system. And an antigen is a macromolecule that reacts with components of the immune system. Then we have something called an epitope. This is a site on an antigen that is actually recognized by the receptor of a cell that belongs to the immune system. One pathogen may have more than one epitope, and each epitope is recognized by a separate specific immune cell. So this is where the immune system has, we had mentioned an analogy before as to the immune system being the Department of Defense of our body, but it is when there is a situation, it's like having an unexpected event, such a fire or an earthquake or a tornado or something, and having the deployment of first responders. And in a situation like an accident, the site of an accident or the site of a fire, you're going to have first responders, individuals that have specific skill sets that are on site, each of them performing a task that is very specific. And that can serve as an analogy with the immune system. And here are some of these specific first responders of the immune system. So some of them are known as T cells, and among the T cells, there are some that are called CD4 or other ones that are called CD8. An important thing is when these cells of the immune system are activated, they have the capability of making copies of themselves. That would be what is considered an expansion or undergoing cell division. In other words, they clone themselves and they can respond against the same pathogen, launching, this self-cloning mechanism. So for the sake of an illustration, here we have a schematic that shows an antigen displaying three different types of epitopes. Okay, and what will become very relevant is the shapes of the epitopes, because these are shapes that will play a role in terms of recognition by cells of the immune system and therefore triggering a response. And in a way, these shapes function in something that is reminiscent of a lock and a key, in the sense that not every lock has a key, but not every key fits the lock, and even keys that fit into the lock, that doesn't mean that they will be able to unlock because of the specific shapes of the key and the lock. So here we have an illustration that perhaps has plenty of information here, but it's valid to see the point in which we have here with a circular shape something representing a pathogen that's invading the body, this pathogen has a site that contains an antigen, and precisely the shape of that antigen helps then to have a binding between something called an antibody. Okay, um, antibodies, we mentioned previously examples of cells that belong to the immune system, CD4 and CD8. Antibodies are also part of the immune system. So let's talk more about responses. We have an antigen that is capable of triggering a specific response by the immune system. Um, there are cells called white blood cells. The white blood cells are, their proper name is T lymphocytes, and they have a receptor called a T cell receptor on the surface. Then we have the CD8. They are also a specific type of T cells. And the CD8s are called cytotoxic T cells. CD8s are also referred as CTLs. And their job is to destroy cells that are infected by virus. The cells called CD4 and CD8 are memory T cells. They expand, they have the ability of expanding um, to large numbers of effectors T cells on re-exposure to their, well, basically what this means is that that functionality of memory is they deploy a trigger a response, they do their job, and then they are capable of retaining information so that if in the future they are exposed again on second or third or continued exposure to the same pathogen, then they would be able to uh, recognize that antigen and epitopes, and they would be able to trigger a response, perhaps even more effectively than the first time around. So this is a very important part of the immune system. Is not just recognizing and launching, fighting the battle then, but is also having the capability of remembering, having this information stored for quite some time, which enables relaunching a fight in the future, if you will. So here we have an illustration of the following. Naive CD4 T cells are undergo activation. They become activated when they engage with cells that are presenting antigens. And they specifically engage a molecule of the antigen presenting cells that is called MHC2. These molecules, MHC, the name of this molecule stands for major histocompatibility complex. And so by doing so, the CD4 T cells become active, and in the process of basically becoming active means that they immediately undergo clonation. They start making copies of themselves and dividing, and at the same time they activate the so-called B cells. This is another type of cells of the immune system, and also they activate or stimulate the CD8 T cells, which are the cytotoxic T cells. And once the cytotoxic, so basically you have the deployment of CD4s, which implies the activation of B cells and also the activation of CTLs. And once the CTLs, the cytotoxic T cells, are out and about, then they're going after locating infected cells and killing them. The CD8s are, they are like samurais or something like that. They kill infected cells, and that's how the immune system, is one of the responses of the immune system. So this illustration here is basically um, having a graphic representation of this process. So in the next part of this lecture, we are going to derive equations that include populations of viruses and include also responses, specific responses by the immune system.